Our next speaker is Dr. Stewart Clark from the University of Buffalo. He's actually a former California citizen, so to speak, and he did his PhD at the University of California at Irvine. And he's talking about a clinical model, a preclinical model in the lab. And Dr. Yu was just right now talking about having animal models in the lab. And Dr. Clark is actually developing a new model of PSP.
Good morning, everyone. Really great to be here. Thank you for having me. So you've heard a lot about the villain, tau. And so my research has taken a little slightly different approach. I'm trying to be the detective that finds out where tau's hideout is. And so tau, we've talked about tau and how it's pervasive in the brain, goes to many different areas. We believe it produces neurodegeneration. But where does tau get its start? You know, tau doesn't just suddenly spring up in all these different brain areas at once. We believe that the disease progress is decades in the making. And so we've tried to think about where this tau could start and where its criminal network gets going. So first of all, why do we need a tau model, or a model for PSP? You've heard from a couple of the other investigators how there's these tau models, and they're great at looking at the villain, how the villain acts in neurons and acts in the brain, but it doesn't recapitulate specifically PSP. It doesn't necessarily go to all the areas that are affected in PSP. One of the main criticisms is that it goes into areas that are affected in Alzheimer's, but are not affected in PSP. So we really need a model specifically for PSP. And why we need this primarily is for drug discovery in two fronts. One is to help reverse or stop the protein aggregation, to stop those villains that have gotten into all these different parts of the brain, but also to improve longevity, but also to improve the quality of life. So as you know, the symptomology for PSP is very unique among these different tauopathies. And so without a model that's specific to PSP, we can't even look for drugs to help with these specific symptomologies. The other thing that we hope to do is to make a progressive model, to take a wild-type, healthy animal and make it have something that's like PSP, so that we can do things like Dr. Boxer was talking about, is look for early biomarkers and look at the disease progression and see what we can learn about early pre-symptomatic PSP. And so what should this model look like? And there's going to be some complicated terms on this slide, but I'm going to go through each one. First, postmortem characteristics of PSP. We have loss of neurons in this. It took me a couple years to be able to pronounce this. I think I pronounce it correctly now. Peduncular-pontine tegmentum. There's also ventricular loss, or ventricular enlargement. We have loss of neurons in this area called substantia nigra, which overlaps with Parkinson's disease. And we have, of course, these abnormal protein aggregates that many of the other researchers have talked about. Behaviorally, I'm going to talk about three things. One is startle deficits, and this is a reflex to loud acoustic noise. Motor deficits that we all are quite acquainted with, and cognitive deficits. So first, let's just talk about the spread. So if we know where the tau goes to, maybe we can get an idea of where it came from, where the hideout is. So early on in PSP, we have it in these areas called the globus pallidus, substantia nigra, and the subphlegmatic nucleus. It also then goes to the thalamus, the caudate, and this PPTG, which I will continue to call PPTG and not the peduncular-pontine tegmentum. But it also goes to the superior colliculus and then goes down into the brainstem and into the spinal cord. Now, as the disease progresses, we get even more aggregates and higher concentrations in these other areas. And it turns out that this structure, the PPTG, sends parts of its neuronal apparatus, what we call axons. It's the long, thin filaments that axons produce to communicate with other neurons. It sends these axons to all of these areas. And so what we decided to do was that because the PPTG it seems to be at the center of this brain network, is that we would basically poison these neurons, disrupt their metabolic systems, and kill them, which does hopefully two things. One is it recapitulates the postmortem from PSP, where here you can see this is actually one of our rats, and it looks just like a normal human and has these neurons in the PPTG. But when we remove these neurons using our metabolic toxin, we remove all of these neurons. So hopefully this recapitulates the disease, at least at this pathological state, and hopefully then it'll precipitate other things. So what we did was we took rats, we removed these neurons using our metabolic toxin, and then we let them live for about 12 to 14 months. And so what we then did was we first looked at that, what I mentioned, acoustic startle reflex. So there's a group, and I believe they're in the Czech Republic, they found in PSP patients that when they're exposed to repeated loud stimulation, normally we all jump appreciably. But PSP patients seem not to. And so this is what we use to do it in rats. There's a loudspeaker here. It's computer control. We put the rat on here. And this is a glorified electronic scale. So when they startle, they press down and we get a measurement. And so in rats that do not have those neurons that I talked about on the PPTG, they don't appreciably startle. So that's a good first step. And this was our very first observation that led my lab on the road to PSP. So then we also did this fun little task we called a horizontal ladder, and it's exactly as you described. So there's this ladder, and we've removed some of the rungs, and we put the rat in the middle here, and they get to choose to go this way or that way. And what we look for is deficits such as paw slips. And so we do this in high def. We have a very good camcorder. And I have my undergrad students score this, and they look for front paw slips. And this is 14 months after the lesion. And in the front paw slips, we don't really see anything. But in the back paws, we see about a doubling of this, showing that they have appreciable motor deficit in their hind limbs. So another motor task that we've done, and we actually developed, as far as I know, no one else does this, we call it the vertical descent paradigm. My students love to call it the drop zone. And so what we have is we have the rat here, as you can see, and they need to negotiate getting from this platform down into their home cage. And rats that have this particular lesion or these neurons removed, they have a whole bunch of different behaviors they like to do, which we call abnormal entries, such as falling into the cage, uncoordinated jumps, flipping in the air, and falling backwards. And so you can see we're a little underpowered here, but we do see an increase in these types of abnormal entries into this new paradigm that we've developed. So the other thing that we also wanted to look at, and again, this is a postmortem study, and so are the ventricular enlargement that a couple different groups have found to be increased in PSP patients compared to both Parkinson's and normals. And now ventricles are these nice spaces of fluid in your brain that basically has seawater in it that bathes your brain, protects your brain. And so these spaces get enlarged in PSP, and that's exactly what we see in our rats. And again, this is 14 months post lesion. And so on the left we have our normal or our sham animals, and then on the right we have lesion animals, and these ventricles are about twice as big in our lesion animals. Okay, again, the substantia nigra, and this is an overlap with the Parkinson's syndrome. So, and we look for what we call TH positive neurons or the dopaminergic neurons. And you can see in our lesion animals, again at 14 months, have appreciable significant loss of these neurons throughout the structure. And I would expect if we let them live longer, they would have even more loss and more motor deficits. So how are we doing on our list? We, of course, started with the pedunculopontine tegmentum loss because we induced that. But that precipitated both a ventricular enlargement, substantia nigra loss, startle deficits, motor deficits. We're still in the progress of looking for cognitive deficits in our animals, and we still probably need to make a better model or using a more longer living animal model organism to see whether we can get a tauopathy. And so what we're going to do is use a viral approach and put in overexpressing tau to see if we can, A, kill the neurons just like we did at the beginning of the study I just talked about, if we can produce abnormal tau in its spread, accelerate in the progressive deficits in behavior that we've seen. And so we've just begun this, and you can see this is in our control virus, this is in our tau virus, and we're getting appreciable aggregates of the tau in these neurons. So future directions, of course, is to complete the tau characterization of both the lesion and the virus models. And we also are going to partner with our friends at Research Triangle Institute to start screening drug compounds in these models. And of course, I do not do this alone. This is the members of my lab who've been through my lab in the last 6 years. And we have some very great clinical and basic science research partners. And then, of course, our funding from Cure PSP. And I'd just like to note, we also started this project on funds from National Institute on Drugs of Abuse. But it was translated into a PSP project. And just so you know, your tax dollars are at work for this project. Thanks.